Hi, Heikki Tanila. You are a professor of translational neuroscience and the leader of the Neurobiology of Memory research group at the AI Virtanen Institute at the University of Eastern Finland. Could you tell me a bit more about what your research group is like and what are some of its key research focus areas?
Hi, Virta. Yeah, I'd love to do that. I think it will be better if I show some slides at the same time. You know, one picture tells more than 1,000 words.
Exactly.
Our main interest is this disease, which name many people start to forget, the Alzheimer's disease, which is the most common reason why we lose our memory in the old days. And we are especially interested in what really makes this disease so specific, that it's the memory that is gone first, and especially it's the memory for recent events. Recent evidence suggests that this is intimately related to sleep. The current idea is that we have some kind of video recorder system here in the hippocampus, but it's also only a temporary storage for the information. So the information has to be transferred to the neocortex, which has large capacity. And this information transfer is thought to happen during sleep. And in order for this to happen, we need three structures: the hippocampus, the thalamus, and the cortex synchronized in a special way. And we know that each of these structures have their special oscillations. High frequencies in hippocampus, sleep spindles in thalamus, and slow waves in the cortex. And it's easy to think that if there's any disease process that disturbs this synchrony, this may impair the information transfer, and therefore we will not make permanent memories for the recent events. We are trying to understand the mechanisms in a mouse that carries those genes that cause Alzheimer's disease in humans. And as you see with the big device, we can have it balanced so that it doesn't weigh too much, and the mouse learns to sleep well in the laboratory. And indeed, in this mouse, we can catch all these oscillations, which I demonstrate here with multi-channel recordings, which is not possible in human patients unless they have electrodes deep in the brain. And now we have a system where we teach the animals to navigate in an open area. And once they have learned that, okay, I go there for an escape, that's the idea of the game, then we have a sleep recording session, and we study how these things synchronize during the sleep. And the next day, we will have a memory test and see if these things are related. Another interesting thing in Alzheimer's disease is that it looks like in early stage, the patients seem to have also epileptic activity, as you see here in these big, small spikes. And we have demonstrated that our mice have exactly the same thing. And one interesting question is that, is this epileptic activity during sleep the thing that disrupts this synchrony? The problem with our approach is that we can only see a small part of the brain at a time. But now we are collaborating with our MRI group led by Professor Olli Grön, and they have developed a new MRI sequence that is special. The problem with MRI is that it's very noisy. Hardly any human, not to speak about mice, can sleep in such a noise. But they have a sequence that is silent. Another problem is that you cannot bring any metal to its large magnetic field, but this sequence tolerates metal electrodes. And we have also already data that we can record brain activity in awake rats. So the question is that why won't we be able to do that in sleeping mice? And that's the endeavor, that we will have electrodes that show that the animal is sleeping, and then we see the whole brain activity. And I think this is the challenge that we now try to address in the current approach.
Wow, sounds fascinating. Do you have any other examples of the kind of research questions you're tackling or the kind of projects that you are doing?
So basically, this is the framework. So we basically want to study the epileptic activity. They are large electric charges which are very difficult to locate. And if we can combine electrodes in the most sensitive area, hippocampus, and then see the whole brain with functional MRI, we could see how this epileptic activity is spread in the brain. And I think this will have by itself many interesting applications in especially neurosurgery, because Kuopio is the center in Finland for epilepsy surgery. Another thing is that we want to understand how these different sleep phases contribute to this memory consolidation. And we are interested in how they are changing this mouse model, but we also do recordings in human patients, and hope that we will, in the long run, be able to predict how the disease is advancing. Is it advancing rapidly or more slowly? And maybe we can tell it just by doing one recording overnight.
Wow, sounds very important. And what are actually some of the achievements of your research group that you are most proud of?
Over the years, I think we have done some significant contribution from basic research to the clinic. For instance, at the moment, we have 3 types of drug treatments for Alzheimer's disease: cholinesterase inhibitors, memantine, and a medical food called Suvenade. And for the 2 latter, we have done preclinical studies that help those treatments to come to the market. And our group was actually the second one in the whole world to detect that Alzheimer model mice have epilepsy.
Okay, wow. And you already mentioned that what are some of the potential contributions of your research, but could you maybe summarize what kind of impact do you wish your research group's research to have in, let's say, the next 5 to 10 years?
I hope that this technique, which is really unique in the world, to combine electrophysiology and functional MRI in sleeping mice that are disease models. No one has done it. And if we get this running, I think it will be extremely helpful in developing, I think, new diagnostic markers, and also to test any new drugs. Because if the problem really is in the sleep, none of the present drugs are trying to do anything to it. So it's a whole new approach to help the disease. I don't claim that we are stopping the disease process, but I think we can find new treatments that greatly help patients to keep their memory alive for a little bit longer.
Sounds very impactful indeed. And you already mentioned that you do some collaborations within the university. Do you have any other kinds of collaborations both within and beyond the university that you have going on?
This project that I was describing, that I do with Professor Grön, is also very closely collaborating with University of Minnesota. There's this Center for Nuclear Magnetic Imaging, which is, I would say, the leading center in this field in the whole world. So we have a very intimate collaboration, so that, for instance, this new MRI sequence that we are utilizing was discovered there, and we are the second lab in the world to apply that.
Okay, wow, amazing.
And then I have some collaboration with a group in Budapest that was the first one in Europe to study what is the influence of this epileptic type of activity to progression of Alzheimer's disease in a clinical setting.
Okay, wow, amazing. Then one last but the most important question. Why should researchers choose your research group for their postdoc? So what makes your research group amazing, basically?
I think for the first, we are a relatively small group. One senior scientist, 4 PhD students, 2 technicians, and variable number of master students. So everybody knows everyone, and everybody collaborates with everyone. Then we are linked to many other groups at the neuroscience community. So I think if one is working in our group, it means that one is also working with 3-4 other groups. And I think our group is special because, my title is Professor in Translational Medicine, I have a medical background, but I'm doing basic research. So it's so easy to also communicate with the clinicians when you know that background as well, which is different from most other groups in our neuroscience community.
Sounds really, really good. Thank you very much, Heikki Tanila.
Thank you, Birgitta, for this time.